Okay, I hope you can hear me. I'm going to go ahead and start with chapter 20. Let me get the share of the PowerPoint up. Okay, chapter 20 has to do with structure and function of the hematologic system. It starts out with the chief functions of the blood, which is to transport oxygen and nutrients to the cells, and all cells require both oxygen and nutrients in order to survive. The red blood cells are the ones that carry the oxygen, and the plasma is the part of the hematologic system that carries the nutrients. They also help discard waste from tissues, protective mechanism by transporting white blood cells where needed, and the maintenance of the acid-base balance. Blood is composed 91% of water, according to the text, although the slide says it's 92%, and the text also says it's 9% solutes. The adult blood volume is approximately 6 quarts or 5.5 liters. And the plasma is the liquid portion of the blood that is the medium that houses all of the elements, both organic and inorganic elements. Plasma makes up about half of the blood volume, and there are two proteins within the plasma component. The first one is albumin. It plays the most essential role in regulation of the passage of water and solutes through the capillaries. It also is, if it's not available in sufficient quantities, blood volume will decrease, which can cause a decrease in blood pressure. Globulins is the second plasma protein. They serve as antibodies and transport substances. And last, plasma contains clotting factors. Clotting factors promote coagulation and stop bleeding from damaged blood vessels. And when we get into the process of hemostasis, that will become more apparent. And among the clotting factors within the plasma, fibrinogen is the most plentiful. There are several cellular components. Erythrocytes is red blood cells. Erythrocytes is red blood cells. Red blood cells are the most abundant cell within the body. Their whole function is to regulate tissue oxygenation by using hemoglobin as the gas carrier. So hemoglobin attaches to red blood cells. And red blood cells, erythrocytes, cannot undergo mitotic division. Therefore, the lifespan of a red blood cell is anywhere from 80 to 120 days. And this accounts for why in a diabetic patient, doing a hemoglobin A1C any sooner than the lifespan of 80 to 120 days is useless, because until the entire lifespan has gone through its timeline, the value of a hemoglobin A1C will not change until new cells are born. Leukocytes are another component of plasma. These are white blood cells. Their primary function is defense of the body. They also act as trash collectors, removing debris such as dead cells. Most of their action occurs in the tissue, but they are transported by the blood. Granulocytes is one of two types of leukocytes. They kill microorganisms. They digest debris through phagocytosis. They act in the chemical response to inflammation. There are three types of granulocytes. Neutrophils, basophils, and eosinophils. And as we learned last week, eosinophils are the part of the white blood cells that attack parasites. A granulocyte is a second type of leukocyte. They have basically the same functions as the granulocytes, except their digestion debris is focused on dead blood cells. And there are three types of agranulocytes: monocytes, macrophages, and lymphocytes. And also natural killer cells are part of the agranulocyte system. Again, their main function is digestion of debris, being a focus on the dead blood cells. And then platelets is another element of plasma. Platelets are not true cells, but they're rather are cytoplasm. Their function is to control bleeding. If there's too many, there will be overclotting. If there's too few, there will be hemorrhaging. And the spleen is the organ, which is the apartment complex for platelets. Basically, the way I remember this is that the spleen will bench platelets until they're needed in the game by other parts of the body to aid in clotting. The spleen is a lymphoid organ. In fact, it's the largest of the lymphoid organs. And the lymphatic system is closely aligned with the hematologic system. There are both primary and secondary lymphoid organs. The primary organs are the thymus and bone marrow. And the secondary organs, as I just said, spleen is the largest of those. But the secondary organs also contain the lymph nodes, the tonsils, the Peyer's patches in the small intestine, and the appendix. All of these organs are sites of residence or proliferation, differentiation, or function of lymphocytes, monocytes, and macrophages. The spleen's main function is to initiate an immune response to blood-borne microorganisms. They regulate hematopoiesis, which is the production of blood cells in fetuses. Now, in adults, hematopoiesis occurs in the bone marrow, but in the intrauterine environment, the formation of blood cells occurs in the spleen. The spleen also serves as a blood storage facility or reservoir with approximately 300 milliliters of blood volume. When the blood pressure drops, the reservoir that the spleen holds is opened, and if needed, 200 milliliters can be rapidly dumped into the venous circulation in order to bring up the blood pressure. The spleen, although not essential for life, and lab analysis has shown that there is an increase in leukocyte circulation when the spleen is not present in the body, and there's also decreased serum iron levels and microscopically damaged red blood cells after splenectomy. So this all builds a case that the spleen does have a function in both proliferation and destruction of these elements. The lymph nodes are cleaners. They clean the lymph fluid of debris flowing through the lymphatic fluid. Their swelling indicates that there is more debris than usual, which is getting backed up. They're the site for the first encounter between antigen and lymphocytes, and they're located throughout the body. They deliver lymphatic fluid back to the main circulation. The mononuclear phagocyte system, or MPS, consists of monocytes which reside in tissue, and in the tissue they destroy microorganisms and other debris. Hematopoiesis is the process of making blood cells. Again, it occurs in the spleen and liver of the fetus, but it occurs in the bone marrow of adults, well, after birth, infants, children, and adults. This process, an increased production of red blood cells, usually occurs in response to chronic long-term stimuli rather than short-term acute issues. Blood cell production, which occurs outside the bone marrow, results from disease such as pernicious anemia, sickle cell anemia, and certain leukemias, which we will look at in depth in chapter 21. As I said, after birth, bone marrow is the primary site for where blood cell production occurs. Bone marrow can be found in the cavities of bones. They host the primary hematopoietic stem cells. In adults, there are two kinds: red marrow, which is the active marrow, and yellow marrow, which is the inactive marrow. And the yellow hue in bone marrow is due to fat. The active marrow is found in flat bones such as the pelvis, the vertebrae, the cranium, mandible, sternum, ribs, the proximal humerus, and femur. In the absence of disease, the blood cells do not enter circulation from the marrow until after differentiation happens. And there are two types of stem cells which are the protagonists to blood cell production. The mesenchymal stem cells turn the osteoclasts, the fibroblasts, and the adipocytes into bone building functions. So mesenchymal stem cells all have to do with bone building functions, and they don't contribute anything to the blood cell production. And then the hematopoietic stem cells are the ones that contribute to the formation of blood cells. And they do that all by a process called, well, no, they don't do that. The formation of red blood cells is done through the process of erythropoiesis. And it requires that the bone marrow be stimulated by a hormone called erythropoietin. Mature red blood cells are termed erythrocytes or normocytes. Reticulocytes are immature red blood cells. In the presence of low oxygen concentrations or hypoxia, the formation of blood cells is compensated in one of two ways. Oops, sorry. In the presence of low oxygen concentrations or hypoxia, the production and the release of erythropoietin is compensated by one in two ways. Either the individual will have an increased respiratory rate in an effort to intake more oxygen from the atmosphere, or there will be an increased erythrocyte production. That makes no sense. Okay, so when the human experiences low oxygen concentrations or hypoxia, the body compensates in one of two ways: an increased respiratory rate in an effort to intake more oxygen from the atmosphere, or the bone marrow, through the process of erythropoiesis, increases erythrocyte production. In patients with chronic renal failure, there may be diminished erythropoietin, resulting in anemia. And I think probably, as we all know, there is a synthetic form of erythropoietin which can be administered intramuscularly in order to stimulate red blood cells in diseases that causes it to be decreased. Hemoglobin is the protein which attaches to a red blood cell and actually carries the oxygen. The heme in red blood cells takes in the oxygen in the lungs and then delivers and deposits it to the tissues. And then while it's at the tissues, the heme will also take out the trash of carbon dioxide from the tissue. So the heme delivers oxygen to tissues and removes carbon dioxide from the tissue. Hemoglobin increases the oxygen carrying capacity of red blood cells by 100%. Hemoglobin consists of polypeptide chains and 4 colorful iron complexes. The iron contributes to the ruby red coloration of blood. In order for erythrocyte formation to occur, there must be several nutritional requirements met: amino acids, the B complex vitamins, folic acid, niacin, ascorbic acid, iron, copper, and vitamin E. And when we get into chapter 21, we'll talk about those anemias that occur because of lack of B complex vitamins, folic acid, and iron, which are the most common causes of anemia. Before we get into the iron cycle, I have just oh, here we go. I don't know why I have them out of order, but I do. So we're going to skip to slide 28 for just a moment. There is a process by which, there's a normal process by which destruction of old erythrocytes occurs. And all of that destruction occurs in the spleen. And if the spleen happens to be absent because it's been surgically removed, then the liver takes over the function of destruction of red blood cells. And then the hemoglobin, which remains when the red blood cell is destroyed, becomes porphyrin, and it's transported to the liver and then secreted in the bile. Now we'll return to the iron cycle. The greatest proportion of the total body iron level is located in the erythrocytes. Actually, approximately 67% of the total body iron is bound to the heme in erythrocytes, and some of it's attached to the muscle cells, the myoglobin. Apoferritin, which is ferritin without any attached iron, can store thousands of atoms of iron waiting to be released. Hemosiderin are large iron storage complexes, so just think of them as warehouses for iron, hemosiderin. And then there are chemical reactions that happen in order for the iron to bind with the iron to bind to the hemoglobin. All of those are oxidative processes. And if for some reason the oxidative process is not successful, then you have deoxyhemoglobin circulating without any oxygen attached to it. So that's it on red blood cells. White blood cells, or leukocytes, also arise from the stem cells in the bone marrow. Granulocytes, which is a type of leukocyte, matures in the bone marrow, but granulocytes and monocytes are released into the bloodstream before they are fully mature. As in the development of red blood cells, white blood cells have growth factors and colony stimulating factors which encourage production and maturation. Just like when someone has bleeding or some type of disease in which there's not enough oxygen being transported by red blood cells, and therefore erythropoiesis increases, the same thing happens with leukocytes, except usually it's an infection of some sort or a steroid depressed, or either steroids have depressed the presence of the inflammatory response, or there's some disorder of the bone marrow which decreases reserves. But when the body feels like it doesn't have enough leukocytes, it increases production. Platelets are termed thrombocytes, and they are derived also from stem cells and progenitor cells. The process by which platelets are formatted is called thrombopoiesis. The megakaryocyte progenitor is programmed to undergo an endomitotic cell cycle, or endomitosis, which results in platelets. About 2/3 of the platelets enter the circulation, and the remainder reside in the splenic pool until they are needed. Thrombopoietin is the hormone growth factor, and it is the main regulator of the circulating platelet numbers. The average lifespan of a platelet is 10 days, and senior platelets are destroyed by the spleen. Hemostasis is simply means stop the bleeding. And there are three components required for stopping bleeding. One is platelets, the other is clotting factors, and the last is that you must have an intact vascular structure held together by endothelial cells. If your veins and arteries are leaking, then it doesn't matter how many platelets you have. There's a series of slides about what each one of these factors do, but I went ahead and summarized it into 5 steps to achieve hemostasis. First of all, when there's an initial vascular injury, vasoconstriction occurs to limit the blood flow. So the vessel starts to shrink down and tighten to stop any blood leaking from within it. Once that vascular injury occurs, platelets arrive on the scene to adhere to the damaged endothelial cells of the vascular wall. This forms a temporary plug in the vascular wall to prevent any additional bleeding. The endothelial cells then secrete a tissue factor which interacts with platelets to form a fibrin clot. And then this fibrin clot contracts even more, forming a permanent plug. But the fibrin clot can't stay in place forever, so a process called fibrinolysis, and if you remember from last week, lysis means to digest. So fibrinolysis occurs to begin to reduce and resolve the clot, which promotes long-term healing. So all these next several slides break those 5 steps down into their individual components. At birth, blood cell counts are higher than they are at any other time in the life cycle. One reason for that is because of the hematopoiesis, which is occurring in the spleen of the fetus, and the other is because of a biological response to birth trauma, both from the delivery process and the cutting of the umbilical cord. Both of those stimulate a response for increased blood cells. Due to frequent viral infections, children tend to have an increased number of atypical lymphocytes. In older adults, the lifespan of erythrocytes remains steady, but the production to replace them after bleeding usually decreases. And that production decrease is usually linked to iron levels. Either the iron level is inadequate because there's been inadequate absorption from the intestines due to motility issues, or there's an alteration in one's ability to achieve iron binding. In older adults, lymphocytes become less functional. The volume may remain steady, but they do not activate. And the hugest impact on the lymphocyte deproduction occurs at cellular immunity, which predisposes the elderly to a variety of the disease states. And then last but not least, the humoral immune system becomes less responsive, and in some individuals it becomes unresponsive. So those are my notes on chapter 20.